Hello, my name is Aidan Skinner, and my presentation will be about determining the degradation of phenethyl isothiocyanate across different insect species. And for the sake of time, I will just refer to phenethyl isothiocyanate as PEI. So we will begin by discussing my introduction. So the microbes associated in the guts of insects have been seen to degrade plant defense chemicals. Pantoia in flea beetles was found to degrade isothiocyanates. Additionally, a species of Pseudomonas was identified as a degrader of caffeine in coffee bear borer beetles. Brassicaceous plants are the plants that are known to house phenethyl isothiocyanates. They also house other isothiocyanates, but we will be focusing on PEI for this presentation. Specialized diets like those found in Colorado potato beetles, yellow margin leaf beetles, and spotted cucumber beetles are more commonly exposed to specific plant toxins associated with their dietary preference. Grasshoppers with a generalized diets are more exposed to a variety of plants, so they intake a variety of plant chemicals. And we sought to identify the degradation ability of symbionts present in beetles, grasshoppers, and caterpillars when they're exposed to PEI. And my hypothesis was that bacteria found in brassicaceous specialists will be better at degrading PEI than generalist and solanaceous specialists. And so we'll move down to methods. And for our methods, our first method was we just collected these specific isolates. And we began by dissecting them, homogenizing their gut. We cultured them, extracted their DNA, PCR amplification. We blasted their data. And then we did Illumina sequencing. And then next we screened the isolates for the ability to detoxify the allelochemicals. And then we measured the impacts of the toxin degraders on the insect growth. So we actually we gave them this toxin for them to consume, and then we determined how their fitness was over time. And so next we'll go to our results. And from figure 1, it is a box plot that shows the bacterial isolates that were labeled as degraders, and they're all grouped up by insects. They're also grouped up by their dietary preferences. So in the first figure, or the first bar plot, you have brassicaceous specialists, and then you can see on the third row, generalists, fourth row, solanaceous specialists. Other just represents the specialists that aren't brassicaceous or solanaceous. So that's kind of just a little extra data. And so next is our figure 2, which is an Illumina plot, and it's colored by the each different column is a different isolate, and each isolate has its degraders labeled. And as you can see, with you can see a vast majority of white space that just represents non-degraders. And so if you look, you can see that on the right hand side, mainly throughout the entire Illumina data plot, you can kind of see that it's being taken over by this blue. That is a Curtobacterium, which is a known degrader. And you can also see there's a little bit of red in there. It's Pantoia. But yeah. And so our summary was, in the results, we saw that bacteria found in brassicaceous specialists are better at degrading PEI. We can also see that most of the bacterial degraders were found in beetles. And the bacteria found in generalist species had a wide range of degraders and non-degraders. This makes sense because they're generalists, so they eat plenty of different plants. And for our conclusion, it was found that 44 of the 96 isolates that were analyzed were found to be degraders, or were found to have symbiote degraders present in their gut. And my hypothesis was supported by my results. It was shown that brassicaceous specialists were better at degrading PEI. And for the future research, these species of insects have been shown to degrade PEI, so our plan is to begin in vivo experiments. And probably before that, we'll also begin to analyze other plant toxins, mainly solanaceous plant toxins, because that'll be our next focus. And we'll probably do a similar experiment with this as this one. And then I would like to thank the NSF Career Grant, which is awarded to Allison Ravencraft. I would also additionally like to thank Jaden Rankin, Tanner Dabber, Amelia Mans, and Casey Martinez for help collecting, dissecting, Sanger sequencing, and preparing cultures. And thank you.